The strength of hormone action refers to how powerful and how long a hormone produces its effect in the body. This strength is not determined only by how much hormone is secreted, but by four major factors: the pattern and duration of secretion, the half-life of the hormone in circulation, the density of receptors on the target tissue, and the affinity between the hormone and its receptor. These factors together control both the intensity and the duration of hormonal responses. Hormones are released from endocrine glands in specific secretion patterns, and these patterns have major physiological importance. In episodic or pulsatile secretion, hormones are released in sudden bursts, usually under nervous control. When neurons in the hypothalamus fire, neuropeptides are released in short episodes, causing pituitary hormones to be secreted in pulses. This pulsatile pattern is essential for normal reproductive function, especially for maintaining a normal estrous cycle. In contrast, prepubertal animals and non-cyclic lactating animals show episodic but irregular hormone secretion. Another pattern is basal or tonic secretion, where hormones are released continuously at low levels with small fluctuations, such as GnRH secretion from the tonic center of the hypothalamus. The third pattern is sustained secretion, in which hormone levels remain elevated and relatively constant for long periods of time. This pattern is typical of steroid hormones because they are produced continuously rather than in response to nerve impulses. High progesterone levels during pregnancy or diestrus are classic examples of sustained secretion. The half-life of a hormone is the time it takes for half of the hormone to be removed from the bloodstream, and it determines how long the hormone can act. Hormones with short half-lives act briefly, while those with long half-lives have prolonged effects. The half-life depends on how quickly the hormone is metabolized and cleared from the body. For example, prostaglandin F2-alpha has an extremely short half-life of only seconds, whereas equine chorionic gonadotropin has a half-life lasting several days, giving it strong and long-lasting biological activity. Hormonal action also depends on receptor density, which refers to the number of receptors present on the target cells. Cells with more receptors respond more strongly to a hormone. Receptor numbers are not fixed and can change due to hormonal regulation, nutritional status, and physiological condition of the animal. Some hormones even stimulate the production of receptors for other hormones. For example, follicle-stimulating hormone increases the number of luteinizing hormone receptors on ovarian follicular cells, thereby enhancing responsiveness. Another important factor is hormone receptor affinity, which describes how tightly a hormone binds to its receptor. A higher affinity means stronger binding and a greater biological response. Because of differences in affinity, two hormones present at the same concentration may produce very different physiological effects. Certain compounds can modify hormone action by acting as agonists or antagonists. Hormone agonists resemble natural hormones and bind to the same receptors, producing similar biological effects. Some agonists may even produce stronger effects due to higher receptor affinity. Antagonists also bind to hormone receptors but produce little or no biological response, thereby blocking the action of the natural hormone and reducing target cell activity. Hormones do not remain in the body indefinitely and are removed through metabolism and excretion. Blood hormone concentrations depend on both the rate of hormone secretion and the rate of hormone metabolism. Rapid hormone turnover is necessary so that hormone action does not persist longer than required. Steroid hormones are mainly metabolized in the liver. During metabolism, double bonds in the steroid structure are reduced, which inactivates the hormone. The steroid is then conjugated with glucuronide or sulfate, making it water-soluble. These conjugated metabolites are excreted in urine and feces. Steroids also enter the digestive tract through bile and are eliminated in feces. Because steroid metabolites are present in feces, fecal hormone analysis is widely used in wildlife and zoo animals to study reproductive physiology without collecting blood samples. In high-producing dairy cows, the liver is larger and more metabolically active. As a result, progesterone and estradiol may be broken down more rapidly, leading to lower circulating levels. This rapid metabolism may reduce uterine support during early pregnancy and contribute to temporary infertility, although further research is needed to fully confirm this mechanism. Protein hormones are mainly degraded in the liver and kidneys. Pituitary gonadotropins such as LH and FSH have short half-lives, typically ranging from 20 to 120 minutes. Chorionic gonadotropins, such as human and equine chorionic gonadotropin, have much longer half-lives because glycosylation protects them from rapid degradation. Human chorionic gonadotropin is filtered by the kidneys and excreted in urine, which forms the basis of pregnancy tests. Protein hormones cannot be administered orally because they are destroyed in the gastrointestinal tract. Hormones can be detected in blood, saliva, milk, urine, lymph, tears, and feces using immunological techniques. Radioimmunoassay (RIA) is a highly sensitive method that uses radioactive hormones and specialized laboratory equipment. ELISA, or enzyme-linked immunosorbent assay, is a safer and more practical alternative that does not use radioactivity. ELISA is widely used for pregnancy diagnosis, disease detection, and on-site hormone testing because it is simple, economical, and reliable. Thanks for watching Vet Care. If you found this helpful, don't forget to like, share, and subscribe for more simple and clear veterinary lessons.